This video is brought to you by Catalyst for Science Policy in collaboration with the Wisconsin Veterinary Diagnostic Lab and University Health Services and University Communications. At Catalyst for Science Policy, we seek to bring the science conducted at the UW-Madison to the Madison and Wisconsin community. You've probably heard a lot about COVID-19 testing lately, and it all may seem a little confusing. Community testing is only one important part of controlling the COVID-19 pandemic on the UW-Madison campus and in Wisconsin. Getting tested is easy. On the UW-Madison campus, we use nasal swab sample collection. The swab only goes in about an inch into your nose, and you can do it yourself. Research has shown that there's no difference in test accuracy in using self-collected nasal swab samples versus those collected by a healthcare worker.
Samples are transported to the Wisconsin Veterinary Diagnostic Lab in these rugged coolers for testing.
We're one of, I think, 10 or 11 different veterinary diagnostic labs around the country that are helping with the human testing in a collaboration with the State Laboratory Hygiene and the UW Biotech Center to make the dedicated COVID lab for UW-Madison. And we decided to go with nasal swabs and PCR. And the reason why we did that is because we wanted a very high amount of sensitivity or low false negatives and a very high specificity, which is low false positives. My name is Keith Polson. I'm the director at the Wisconsin Veterinary Diagnostic Lab. Our expertise is high throughput testing, so we manage large epidemic outbreaks on a regular basis. Lots of samples, thousands per day, with a very high quality.
Scientists at the WVDL have been working tirelessly since the spring to set up their lab to run human COVID-19 diagnostic tests. Their impressive workflow is made to ensure that their tests are reliable and that you get results back within 36 hours.
Within the coolers are the individual sample bags that have been collected by UHS. My name is Dan Christensen. I am the Client Services Interim Section Head at the Wisconsin Veterinary Diagnostic Lab. We do a first check of looking for any leaking. One of the requirements for CLIA testing is that we have an order for each sample we receive. So the unique identifier, which is unidentified barcode number, has to match a manifest list that we get from UHS each day. We scan the barcode, which is a QR code, into the spreadsheet, and you can see here that it matches the manifest data. So we know that we have an order for this specimen. The next thing we do is upload this information into our LIMS system and generate individual barcoded labels that will have the same unique unidentified identifier on it, along with our LIMS position and accession number. And then we will adhere those to the samples one by one, double checking the ID, positive identification of specimen, which is also checked by the testing section for a second check. Once all the samples have gone through this process, they are ready to be transported up to the testing lab and ready for the testing procedure to begin.
When the samples come in, they're going to be put on a machine very similar to this. These are automated liquid handlers. So this is a way that we can do this with 8 microbiologists to get several thousand samples through per day. When you think about the high throughput and the number of samples that are actually coming through the lab, just the ergonomic issues and the error rate, if we were doing them one-to-one, would be extremely high, and we would need a lot of people to do it.
The second step of the COVID-19 test is nucleic acid extraction. This step is very important for the overall sensitivity of the test. The virus contains genetic material called RNA, or ribonucleic acid. This is a single-stranded molecule that contains instructions for the virus. The process of nucleic acid extraction simply involves mixing the samples in the 96-well plate with special chemicals to extract the genetic material from the cells in the sample.
So what this does is in 96-well formats, instead of taking a pipette individually, it rotates that 96-well plate around and it extracts the RNA.
We then reach the third and most important step of the COVID test, the real-time reverse transcription polymerase chain reaction, or real-time RTPCR. Several chemicals are added to the extracted genetic material, which is then loaded into an instrument called a thermocycler. Throughout the experiment, the thermocycler cycles through different temperatures and detects an output signal. But what actually happens during RT-PCR inside the thermocycler? Reverse transcription, or the RT, refers to the first step in the process, which converts single-stranded RNA into double-stranded DNA. The polymerase chain reaction refers to the next step of the process, which goes through many cycles to replicate only the viral DNA sequence of interest. The thermocycler heats up to 95 degrees Celsius, causing the strands to separate. When separated, important bits of cell machinery come together to build a complementary strand alongside each original strand. Each cycle, therefore, doubles the amount of DNA present. As a part of the cycle, a probe molecule binds to the specific DNA site and later gets broken down, causing it to fluoresce. As the number of DNA strands increases exponentially into the billions, the fluorescence also increases, and the instrument detects that signal. Therefore, an increase in fluorescence indicates that the sample is positive for the presence of the virus. Remember, the amplification process only occurs if the particular SARS-CoV-2 DNA sequence is contained in the sample. Testing is just one critical component of Wisconsin's strategy to reduce the spread of COVID-19. For health advice, always refer to your doctor, the CDC, and your local public health department.